Welcome you all again. Last lecture I discussed about resolving power of any microscope as well as the history of the electron microscope. Now let us discuss about the different components of the electron microscope and how this electron beam is interacting with our sample, and when it is interacting with our sample what kind of electron beam we are expecting. So, let us take a look to our signal generated when a high energy beam of electron interacts with a thin specimen. So, here this is our thin specimen and this is our incident high voltage high kilovolt electron beam. I will just use this is a incident beam, and then when it will interact with the sample there is a two possibilities, or you can say there is a three possibilities. One is electron beam will interact with the sample and sample will absorb some electrons, and that is basically damaging the sample. So, that is very important part for the biological sample, because a biological sample is made by only amino acids or DNA, RNA, protein, and if there is a very strong electron beam which has a lot of energy, it is going to damage that protein structure or DNA structure. So, if electron will absorb, that is bad for the sample. So, if sample is not very thin, chances are very high incident electron beam will interact and lot of energy it will absorb, and that will damage the sample. So, one more thing. So, here whenever we are imaging biological science sample, we cannot expose the sample for long time. So, there is a short time exposure is there, we have to use that. Now, when the incident electron beam it will come, it will absorb, but not 100%. Some of the electron beam will transmit. That is why we are talking about the transmission electron microscope. The direct electron beam it will come. So, some electron beam will be transmitted. When it is transmitted, there is a two possibilities are there. It may interact with the some atom of the molecule and lose their energy, and then it will be a inelastically scattered electron. And the other way, if there is a no such kind of interaction, only the deviation of their path length, there will be a elastically scattered electron. Now, here we have to remember that whenever we have an electron beam, we are always considering that electron beam is a coherent electron beam. So, this coherent electron beam, we are expecting if there is no energy loss of that electron, that will come out from the sample and it will be a coherent elastically scattered electron. Other possibilities is that if we have a only photon light, so lot of time light will, if there is a mirror or glass, so light will come and reflect, right. Same kind of phenomena is possible here also. When incidental light it will come, it will go back, and that time it will generate the secondary electron or back scattered electron, other electrons, and sometime it will generate some X-rays or some visible light. So, we can use a different kind of attachment where we can record that what kind of rays it is coming out. From there we can discuss, we can figure it out what components is there, but those particular things mainly we use for the material science purposes, not for the biological science purposes. But biological science purposes, the secondary electron and back scattered electron, and here all these electrons are very important. So, we are going to discuss about those things now. So, we are always considering that when there is a electron beam is coming from the electron gun, it will be always a parallel electron beam. So, that is the incidental electron beam, and it should be a parallel, there is no deviation. When there is a thin sample, it will interact with the thin sample, and then there will be a change in the scattering with a varying intensity. Why? So, that we will discuss more in the next slide. But when this electron it will come, we can get the so there is a incident electron beam which will go through and there will be no interaction, and if it will interact it will give us a diffraction pattern and forward scattered beam direction. Now, whatever I was discussing that okay, there is a coherent electron beam. So, this is the coherent incidental electron beam. As I mentioned, we are not interested about the spectroscopy, that is why we are removing our X-ray and the visible rays from our discussion. Only we are going to discuss this backscattered electron, secondary electrons and the different type of transmitted electron beam. So, we have a coherent incident beam. The coherent incident beam will pass through the thin sample and this is the direct beam. There is no scattering, nothing. Now, when there is a scattering, as I mentioned, there is a three possibilities are there. One possibility is that it will directly interact with the electron and lose their energy, and other possibility is that it is very close to the electron, but there is no interaction. When there is no interaction, there will be no loss of energy. So, we can expect there is a coherent elastic scattered electron, incoherent inelastic scattered electron, incoherent elastic forward scattered electron. Other two, it will be backscattered electron, that is incoherent elastic backscattered electron, and secondary electron from the specimen. If there is a very thick sample, what I mentioned that the coherent incidental beam, it will come and then it will bounce back or reflect back from the sample, and then we will get the secondary electron, we will get the incoherent back scattered electron beam, but and also incoherent elastic backscattered electron beam, but we cannot get any transmitted electron beam because sample is very thick. So, here we are learning one most important part, that if we want transmitted electron beam, means transmission electron microscope, we need a thin sample. If sample thickness is very high, we cannot use transmission electron microscope. Second one, that here you can see, previously I discussed that backscattered electron and the secondary electrons are very important for scanning electron microscope. That is the reason for the scanning electron microscope, if sample thickness is high, it does not matter, we can get some kind of backscattered electron beam or secondary electron beam to image the surface. Here you can see incidental electron beam is coming and reflecting back from the surface, and that is basically we are recording when we are doing the scanning electron microscope. Now, let us take a look what kind of interaction we are expecting when we are imaging some biological science sample. Okay. So, here we are we want to see what kind of interactions are there between the incoming electron and the atom. So, let us draw atom first. So, any atom, it will be proton and neutron at the nucleus. So, it will be closely packed something proton and neutron. So, if we just consider this is nucleus and this should be our first orbital, then it will be a second orbital and then third orbital, and electron will be everywhere. So, if we just consider this the s orbital, p orbital and d orbital. So, I am not trying to fulfill all these orbitals, then it will be very difficult for me to draw that scattered unscattered electron beam. So, here first thing first, we consider that backscattered electron. What is happening? This is a incidental electron beam which is very close to the nucleus. It will come and deflect its path and then finally it will go out from this atom completely. This is called the backscattered electron. There is another possibility that okay, one electron is coming here. So, this is the backscattered electron which is very close to the nucleus and reflect back and giving us some kind of. So, this kind of backscattered electron, it may interact with the other electron and lose its coherent property. So, what is the coherent electron beam when we are talking about? So, it is good to know that coherent electron beam. So, if there is a electron gun, we are always expecting that all the electrons are starting from this point. It should not come from any other point. So, if all the electron beams has the same energy, same path length and same wavelength, it will be follow this kind of where the amplitude, phase, wavelength, everything is same. So, phase, amplitude, lambda, path, everything is same. Then we are telling that this is a coherent electron beam. So, electron beam. Now, incoherent electron beam means if we look at a rays is coming like that, and the other hand there are some rays are coming this way, some are coming like that, and the other hand couple of electrons. So, these electrons are coming from it. So, this is called the coherent electron beam. Don't forget, this is the coherent electron beam, and here we are drawing that. So, there is a 2, 3, 4 waves are there, where one wave is moving in this direction and other wave is moving in this direction. And if you look at this position, they are completely different phase. They are opposite to each other. Same thing here also, they are opposite to each other and they are going to normalize each other. So, this kind of electron beam is called incoherent electron beam. So, we can always consider that when electrons are coming from the electron gun, so electron will be most of the time coherent until it will interact with the atom. So, we will just consider that okay. So, before interacting with the sample, all the electrons beams are the coherent electron beam. In practical case scenario, that is not the case, but we are just considering for now. So, all the electron beam will be coherent, but when it will interact, there is a possibility some electron beam will be coherent and some it may interact with the electron or nucleus of the atom and then lose their energy. So, whenever electron will lose their energy, their wavelength will change, their path direction it will change, their phase it will change, and electron beam will be non-coherent electron beam. So, now here one electron it is coming, and if it is directly hit here, there is a two possibilities. If there is a high energy and high speed electron beam, it will just displace this electron beam in this direction. So, this red electron it will come out from the path and it will go to this direction. So, this electron it is called the secondary electron. So, for the scanning electron microscope, we are mostly using these two electrons. Now, if there is a electron, it is coming here and it hit somewhere here, okay, and this electron beam, this electron is displaced from its actual position and it is going to other orbital either here or it is going to another orbital here. So, this case it does not has that much energy, or it cannot displace this electron from this atom and throw it completely out. So, if this electron here, if it is not displaced completely out, if it will jump from the lower orbital to the higher orbital and it will release some kind of other energies, it will release. So, that energies it is called that X-ray or visible ray. So we are not very interested about this particular part. Let's see the other scenario, what is happening. So there will be some electron, it will just pass through. There is no intention to interact. So this electron has the coherent electron beam, and this coherent electron beam is going as it is. So these electrons are called unscattered electron beam. Okay. Now other possibility is that, okay, one electron is coming very close to this electron, and because of this electron there is a repulsive force, and this electron will deviate either this direction or this direction. So what is happening, this incident light, it is going straight, it is not too close to nucleus or any other electron, and that is the reason this electron is not scattered at all, and that is called the unscattered electron beam. But if the incoming electron is very close to other electron, there may be a deviation in their path length. This path length may deviate in this direction or this direction. So here you can see there is no energy loss, there is no direct electron to electron interaction, it is just a deviation of their path length. So that is the electron is called scattered electron. So this is called the scattered electron. So this electron scattered. Now what happened to this kind of electron where there is a direct interaction and it replaced this electron from here to here, or this electron is completely kicked out from this atom, and then we are getting the secondary electron. But what happened to this incident electron? Okay, so this incident electron, we want to draw that incident electron here. So we will erase this part and then we will see. Now in this particular case you can see that electron is coming here, it interacts with this electron, and then definitely due to this collision there will be some energy loss. What will happen? This electron will be also scattered in a different angle and different direction, even this one also a different angle. This is called inelastically scattered electron. So most of the cases those are elastically scattered electron. These scattered are called the elastically scattered electron because there is no energy loss and it is maintaining that amplitude, path, phase, and they are coherent. But here due to this energy loss, this is the situation this electron is facing, where there is a difference between their phase, path length, wavelength, and they are basically inelastically scattered electron beam, which is basically creating noise in our image. So the scattered electrons are always good, and that is giving us a good signal. So in electron microscope, whenever we are talking about, there is a two types of contrast we are going to discuss. One is amplitude contrast and another one will be the phase contrast. This phase contrast is coming from the scattered electron that we are going to discuss in the later stage. And unscattered electron beam and the scattered electron beam both are good, but this inelastically scattered electron beam, where they have a different wavelength and path and the different phase, that is basically adding noise to our images. So now I hope it is clear from this also that why we are talking about the coherent elastic scattered electron beam, and the other cases when there is a electron collision here, electron and incoming electron collision, direct collision here, you can get it in these cases, you can get it incoherent incoherent inelastically scattered electron, and some cases the beam is incoherent but there is no direct collision. If you just consider that electrons are very close to each other, there is a chance there is some changes are happened, beam are not coherent anymore, so waves are going in a two different, but it is a elastically scattered. So here you can see that why some electron beams are incoherent and some are coherent, why some electron beams are elastically scattered and some are inelastically scattered. And when there is a thick sample, similar phenomena are also here, that elastically scattered or inelastically scattered electron beam, and so coherent and incoherent electron beam. So we have to consider all of these things when we will think about any image formation. And biological science sample, always remember that. So for the major difference between the material science electron microscope and biological science electron microscope, we cannot use very high electron dose for the biological science sample, because as I mentioned, if your sample is very thick, or any kind of biological science sample, it is not very shiny surface that it will reflect mostly, and then it cannot tolerate electron beam for long time. So if it will absorb, it will damage our sample. So that is the reason we have to use a very less amount electron dose to keep all this soft material properly when you will do the imaging. That is the another difficulties, because we will not get the enough contrast. So that we are going to discuss in the later stage. So but for the material science, we can use a huge amount electron dose that is not going to damage the sample. So that is one of the major difference between the material science sample and the biological science sample. Now we are talking about the transmitted electron microscope. Here there is a photograph where you can see there is a two rhinos are there, but this photograph is taken in such a way so here you can see there is a two rhino, and both the here all the four legs are visible, and the second rhino another three legs are visible, but this photograph is taken in such a way it is looking like it is a double headed rhino. So one rhino and the back side there is another head, but this is not the case. This is some kind of, due to the photography, this artifacts is generated. So here also you can see this kind of artifact when we will do the electron microscopy, especially transmission electron microscope. Why? Here is the example that, okay, just imagine that this is a sample. Okay, so just consider this is a sample, and there is another sample is this. So now if they are separated from each other, so this is a 3D object and electron beam is passing through that sample, and we are getting a 2D image of that 3D object. So in 2D image you can see that, okay, there is a 2 hand you can see and 5 fingers you can see properly. Now if this sample is overlapped with each other, and when the electron beam it will go through, you cannot see all the 10 fingers, you can see only 4 fingers, and it is looking like that this fingers is attached with my right hand. So there is this kind of possibility when you will prepare the sample, it may overlap, and that can give you a wrong images, or it is not giving a wrong images. When we are analyzing our data, we are analyzing it in a wrong way. We are not understanding how this 3D object is going to form a 2D image, and that is the artifact. It can come from the transmission electron microscope as well as from the cryo-electron microscope. So we have to be very careful when we will do those image analysis part. Now different types of electron microscope, that is I already discussed that transmission electron microscope, scanning electron microscope, then STEM is also there, scanning transmission electron microscope. And now if you just consider this transmission electron microscope, you can consider for the material science purposes, you can tell that analytical electron microscope or high resolution TEM or cryo-TEM also. So, cryo-electron microscope we are using at cryogenic condition. Similarly, the ACM also has a different types of ACM that you can use it at cryogenic condition, you can use for environmental ACM. So, different types of ACM and TEM is there. We are not going to discuss all of them. So, we will just discuss about the basic principle of scanning electron microscope and then we will go to the basic principle of the electron microscope. So, here this is the architecture of a scanning electron microscope. What is this architecture? Here you can see this is the electron gun, or you can say this is the source of radiation. For light microscope we are talking about light, this is the similar thing like light. So, it is the light source of our microscope. So, that in electron microscope we are telling that it is called electron gun. So, this electron gun has a different types of component that we are going to discuss in details in next class. So, here you can see these electron beams are coming here and converge in one particular point. So, we can see a condenser lens, there are two condenser lenses are here and one objective lens are here. So, any types of electron microscope you can see this kind of architecture where you have a two condenser lens system and then one objective lens system. In transmission electron microscope it will be always, it will come like condenser lens, then objective lens and rest of the lens it will come. But here our sample is somewhere here where the electron beam it will come from the condenser lens, then objective lens and interact with our thick sample, and then there is no need to transmit this electron through the sample. So, sample it will backscatter electron or secondary electron. So, here we are just recording this backscattered and secondary electron using an amplifier, and that we are generating that signal and we are recording that and we are visualizing those images. So, here it is written that mostly that secondary electron with the most probable exit energy is 2.5 electron volt and by backscattered electron. So, there is a two electron source, one is secondary electron and another one is the backscattered electron are helping us to give us that image from the scanning electron microscope. What about then electron microscope, transmission electron microscope? So, transmission electron microscope we already discussed about that. Most of the time it will be 120 kV or 200 kV or 300 kV. kV means kilovolt. Now, this is the structure of that transmission electron microscope. Generally it is 7 to 8 feet long. So, it is not a very small tabletop equipment. It itself has a table. Here you can see a table where there are multiple control panels are there. So, generally electron microscope sizes are very big. So, like when I was discussing about the history of the transmission electron microscope, you saw a large cylindrical column kind of shape is there. Here also you can see a cylindrical column, similar architecture where we have the filament or gun, electron gun at the top, and there are multiple other parts are connected with this cylinder where we have different kind of lens system, as I mentioned that condenser lens or objective lens. Now, we are coming to that particular diagram, we want to see it inside. So, someone can tell that okay, in light microscope we can see objective lens, we can see sample holder region, we can see condenser lens and other lens system, even we can see the light source. But here everything is under a cover, under a cylinder. So, here you can see this is the viewing chamber where you can see this green light where all these images are forming, and we can also get all these images in this computer. But why we cannot see all this lens system? Why we cannot see the electron gun? We cannot see all of these things because this entire system is under a high vacuum. If you do not have a high vacuum, electron beam cannot come and it cannot illuminate your sample and it cannot pass through your sample. It will interact with the other molecules or other atoms or air and it will deflect, and you will get only noise. It cannot generate a good amount electron and scattered electron beam to give us a good image. That is the reason we cannot see that electromagnetic lens or gun or other parts, because entirely it is under a high vacuum. Not only that, even our filaments are very delicate and it is a very expensive filament. So, if we try to generate that kind of high heat in a open form, that filament will be damaged, our lens will be damaged and aperture will be contaminated. So, there will be so many other parts are also there. I am not going to discuss this. When we will discuss about that, the vacuum system of the electron microscope, that time we will talk about that. Now, if you pay attention to internal part of this microscope, you can see it. It has a electron gun and then it has an anode. Then there is a 2 condenser lens or sometimes 3 condenser lenses are there. Here you can see condenser lens 1, condenser lens 2. And then specimen is going somewhere here. This is the goniometer or specimen holder. So, specimen is going here, where there is a 2 objective lenses are there. One is called the upper objective lens and the lower objective lens. This, when electron beam will go through the 3D object, it will pass through this objective lens, then intermediate lens, projector lens, and it will give us an image here in the fluorescent screen. So, let us take a look how the electron beam is going through our sample and give us an image. So, here we are getting, so electron beam is passing through the sample and we are getting a 2D image. So, here we have a 2D object, 3D object here, and 3D object is giving us a 2D image of that 3D object. So, 3-dimensional view, it will be not that clear from these 2D images. As well as if we want to do the surface study, this transmission electron microscope is not a good tool to do the surface study. Surface study we can always do using a scanning electron microscope. So, something like that, we want to give an example that we have a cell. So, RBC or neutrophil or macrophages. Now, we want to incubate these cells with the toxin and I want to see the morphology change of the cell. Whenever we will add some bacterial toxin or viral toxin, it will change the outer membrane of the cell. So, when it will change the outer membrane of the cell and I want to see the surface change of that cell, definitely we should use the scanning electron microscope, not the transmission electron microscope. Transmission electron microscope will not give us that clear picture of that surface, but if we incubated the toxin, toxin is going inside the cell and it is damaging some organelles, and now I want to see how it is damaging inside the cell and I want to section that cell and want to image, that time we can definitely use this transmission electron microscope and image. So, there are different types of electron microscopes are there. First, we have to understand what is my objectives, what I am trying to address, and based on that I have to decide which equipment I am going to use it. There are many cases you do not need to go cryogenic condition at cryo temperature to do the transmission electron microscope. In that particular case, you can just use the normal room temperature transmission electron microscope. So, this is a microscope what is it can easily operate at room temperature. So, this is we can prepare our biological biological sample at room temperature and we can image it. So, we do not need very cryogenic temperature or condition. So, based on our criteria we can select our equipment. So, at this moment I will stop today here and we will go in details about that electron microscope, different component of the electron microscope, electron gun, electromagnetic lenses, and then different types of aberration and image formation in next class. Thank you all for your attention.